Now, let's start with recombination. That's a fresh chapter. So, we are going to first take a broader meaning of the word recombination. And after that, we will be coming to that meaning of recombination, which brings it to crossing over. But first and foremost, we are going to see the broader meaning of recombination. So, a lot of biology students are not very clear about what is recombination. So, first and foremost, let's get clear about that. So, basically, what happens in recombination, that genetic information that earlier did not exist together, now if it is existing together and it is expressing, that is called recombination. So, for example, before you took birth, there was no other individual which had some characters from your mother and which had some characters from your father. So when your father contributed one set of chromosomes and your mother contributed one set of chromosomes, all right, and of course, presuming that you have no sibling. So when these one set and one set of chromosomes came together and they made one complete diploid set of chromosome. So what happened? That two such sets of chromosome came together in the same nucleus, which were earlier not existing. And now you have a bundle of features from your father and a bundle of features from your mother. And who you are? You are a recombinant between your father and your mother. Simple as this. For certain features, you are more like your mother. For certain features, you are more like your father. So this is a simple picture, though you might show some of your grandfather's character and all that, but we are not going there. Now that is one type of recombination. Other type of recombination is that there is some close range interaction. Mind it, there is some close range interaction. मतलब दो DNA हैं, they are very close with each other, और उनके बीच में there is some interaction. Okay, between two DNA, and then some amount of genetic information moves from one DNA to another DNA. This movement can be reciprocal, मतलब कि ये दोनों तरफ होगा कि थोड़ा सा इंफॉर्मेशन मैंने उसको दिया, थोड़ा सा इंफॉर्मेशन उसने मुझे दिया. So this can be an exchange of information, that is, it moves both way, it is reciprocal, or it can be one way also. Whatever the case is, this is also a type of recombination. But if you look very closely, then in one type of recombination where you are a recombination of traits of your father and mother, right? In this case, the two set of chromosomes have come from two different sources and now they belong to the same nucleus. In this case, there was no such breaking of chromosome fragment going into another chromosome or getting exchanged with any other chromosome. That is not really happening. This is called interchromosomal recombination. Interchromosomal, that is between chromosome. We also call it nuclear recombination, means this is a recombinant nucleus. It is neither exactly like your father nor it is exactly like mother. This is recombinant nucleus. In Mendelian crosses, when you cross pure tall with pure round seeded plants with dwarf and wrinkled seeded plant. So in F1, all of them are going to be tall plants with round seeds. But if you carry out selfing, what happens in F2? You definitely find tall plants which form round seeds. You also find the dwarf plants which form wrinkled seed. But do you notice you are also finding two categories of recombinant? What are the two categories of recombinant? The first category of recombinant where the plant is tall but the seed is wrinkled, and the other set of other set of recombinant is where the plant is dwarf but the seed is round. So these two categories are very much recombinant, but in this case simply new combination of chromosomes have been created and now a recombinant individual has been produced. So this is called Mendelian recombination, nuclear recombination, or interchromosomal recombination. In this case, the DNA does not exchange segment with other DNA, or DNA does not introduce a segment into another DNA. But then other type of recombination is crossing over. In this what happens? Two homologous chromosomes during the period of meiosis, they come together. Certain enzymes are there. These enzymes make two homologous chromosomes exchange equal amount of genetic information with each other between non-sister chromatid. This is also recombination, but this recombination is going to be called intrachromosomal recombination. Intrachromosomal primarily because it is involving breaking of chromosome, and inside of the chromosome recombination is occurring. There are two types of intrachromosomal recombination. Number one is reciprocal, where there is equal exchange of genetic information, and second is one way. The greatest example of one-way recombination is site-specific recombination, where one piece of genetic information inserts into a target DNA at a specific site, and that is called site-specific recombination. So the other type of recombination is called molecular recombination or intrachromosomal recombination. This kind of recombination breaks linkage, right? The first type of recombination does not impact linkage, but yes, it will create offsprings which are different from the parent. So how do we define genetic recombination? The definition of genetic recombination will be bringing together the genetic information that did not exist together earlier. That did not exist together in where? In the parent. पेरेंट में नहीं थे, लेकिन अब वो जेनेटिक इंफॉर्मेशन एक साथ है. एज अ रिजल्ट what you get is recombinant offsprings. What do we mean by recombinant offsprings? The offsprings we have already done, so I don't need to explain this. Write this, explain to kar hi raha hu. What we mean by recombinant offsprings are the offsprings which are neither exactly like one parent nor exactly like other parent. They have some features from one parent, they have some feature from the other parent. Okay? So that is recombination. I hope you are clear. Now we come to the definition. Recombination is a genetic process which produces an offspring with unique combination of traits which differs from either parent. Another way of saying the same thing, that bringing together of genetic information that did not exist together in the parent. The result is recombinant offspring. Other way to look at it, genetic process which produces an offspring with unique combination of trait which differs from either parent. Example, let's say that in Mendel's experiment, so tall plant with wrinkled seed was one recombinant, dwarf plant with full or round seed is other recombinant. But in Betson and Punnett, who discovered linkage, correct? In Betson and Punnett, so you saw red flower plus long pollen as one set of recombinants, and purple flower and round pollen as other set of recombinants, right? But this is result of what? Crossing over. Why? Because they are linked genes Are we all clear? Can we all recall that they are product of crossing over since the two genes happen to be linked? Rather, in Lathyrus odoratus, this was the trait with which the phenomenon of linkage got discovered. Just recall from what we have done in day before yesterday's class. And this is what? This is result of independent assortment. So can you see here that in both the cases, we see that offsprings differing from either of the parent are getting produced. Offsprings differing from either of the parents are getting produced. But in one case, it is just creating a new combination of gene alleles, right, at interchromosomal level, or this is nuclear recombination. Nuclear recombination or interchromosomal recombination. And what is this? This is intrachromosomal recombination. So two examples in both of which offsprings are produced which are unlike any of the parent. If there is any point which is not clear, please revisit the Mendelian cross, please revisit the Betson and Punnett cross in the beginning of linkage, and it will be amply clear what I am talking about. So this is what I was telling you, that there are two modes of recombination. If the genes happen to be non-linked, like Mendel was seeing, in this case, independent assortment is the mechanism that will produce recombinants. So genes are located on different chromosomes, and during meiosis, they will go through independent segregation that is known as independent assortment. This is something that we have already done in Mendelian chapter. This is known as interchromosomal recombination or nuclear recombination. What is the other type? Linked genes. So the genes are located on the same chromosome. So when the genes are located on the same chromosome, then it will involve breaking of DNA and exchange of genetic information. This is what we are going to do today. This we call intrachromosomal recombination or molecular recombination. Now, when you talk about molecular recombination, there are two dimensions. Molecular recombination can be reciprocal and it can be non-reciprocal. Okay, so there are going to be two types of molecular recombination. Reciprocal. In reciprocal, there is basically an exchange of equal amount of genetic information. And then there is something called non-reciprocal. All right. So this is one way. So let's imagine that there is this genetic information and this genetic information gets into this one. So this genetic information plus this genetic information. So what we are likely to see is this. So this is what? This is one way, right? There is no exchange. But then you also know that during meiosis, prophase, and that too prophase 1, there will be exchange of genetic information between homologous chromosomes if they are crossing over. And the fact of the matter is that crossing over takes place. Crossing over takes place pretty much between every single homologous chromosome pair. Crossing over is very 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 common occurrence. हर पेयर में होता है, लेकिन हाँ ये जरूर है कि आपके बताए हुए दो जीन के बीच हो रहा है या नहीं, दैट इज समथिंग which is not guaranteed, but every chromosome definitely goes through at least one crossing over. So once this crossing over has happened, so at the end of it, what you are going to see is that the purple chromosome gets yellow segments and the yellow chromosome gets purple segments. That I am going to draw in a second's time. So this is essentially an exchange process. So now if you see, so yellow chromosome has got some purple segment, and exactly for same amount of DNA, the purple one has gotten yellow chromosome segment. So in this one, in non-reciprocal, all right, in non-reciprocal, new genes get introduced. In this one, alleles get exchanged. Because these are two homologous chromosomes, so eventually they are going to have the same gene but different alleles. All right, same gene but different alleles. But when it comes to one way, then this blue segment is indeed having some new genes. If it is having a gene, then that will get introduced in the yellow DNA. Uh, Anushka is saying, why don't we name E.D. Saunders as well along with Betson and Punnett? We can very well use it. Like I'm more habituated of talking about Betson and Punnett, but yes, E.D. Saunders was also there. So you can very well mention. Why not? Right? Why not? So these are the two types of molecular recombination. So now the same thing in this presentation. So there are two types of recombination, nuclear or interchromosomal recombination. It arises from sexual reproduction where non-parental combinations are created. No change in the chromosome structure, also called the Mendelian recombination. This is your example. Round and yellow seeds, wrinkled and green seeds. First generation, all round, all yellow, but carry out self-fertilization and you will get round and yellow like one parent, wrinkled and green like other parent, but round and green is something that you are seeing afresh and wrinkled and yellow is something you are seeing afresh. So this is Mendelian pattern of recombination. In this, chromosomes have not gone through any structural change. Now, this is molecular or intrachromosomal recombination. Exchange of genetic information between DNA molecules. It gives rise to new combination of genetic information on one DNA. Wait a moment. On one DNA. And it changes the syntenic relation among the genes on a DNA. Now the question is, what is synteny? Synteny. So the syn will mean together. Teny means thread. So synteny means together on a thread. So let's say that if on a chromosome gene A is dominant, gene B is recessive, gene C is dominant, gene D is dominant, gene E is recessive, gene F is recessive. So this is a particular type of syntenic relation. Imagine that there is crossing over and this F changes into dominant F. Is there not a change in syntenic relation? Of course there is change in syntenic relation. Or let us imagine that between A, B and C, there is a new gene, one way it has entered by site-specific recombination, one-way process, a new gene P. Did it not change the syntenic relation? Of course, earlier it was dominant A, recessive A, recessive B, dominant C, dominant D, recessive E, recessive F. Now, crossing over can change perhaps this recessive F into dominant F, syntenic relation has been changed, or one-way process can introduce a gene somewhere in between B and C, C and D, wherever. That also changes syntenic relation. So in molecular recombination, syntenic relation gets changed. In molecular recombination, syntenic relation gets changed, whereas in Mendelian recombination, there is no impact on syntenic relation. I hope you are clear about this also. Here is crossing over. Homologous chromosomes are aligned. Now you can see very nicely the gene alleles are shown. So on the blue chromosome, A, B and C are present in what? Dominant form. On the red chromosome, the same three genes are present in recessive. There is a crossing over happening here during meiosis prophase 1. And now do you notice here that gene C has been exchanged. So now on one of the chromatids, A and B are dominant, but C happens to be recessive. And here A and B on red chromosome, on one of the chromatid, A and B are what? Recessive, but C is dominant. So now these two chromatids are non-recombinant, whereas these two chromatids are recombinant. So that is crossing over reciprocal. Gene C was exchanged between them. But notice here, site-specific recombination. This is a real-world example of a bacteriophage. So lambda bacteriophage injects its genetic material into the E. coli bacterial cell. And this is the E. coli bacterial DNA, and the lambda genetic material in the red color will get integrated into the genetic material of Escherichia coli, and that too very, very precisely between two gene operons, galactose and biotin operons. So between these, it will be inserted. So bacteriophage DNA is inserted into bacterial DNA at a specific site between galactose and biotin operons. So this is site-specific recombination. And one is called transposition. So transposition is insertion of a specific piece of DNA inside some other DNA. So there is this a specific piece of DNA, it will simply go and it will insert into other DNA. But it can insert at any site, number one. And in this there is an enzyme responsible that is known as transposase. Therefore we call it transposition. There are two types of transposition. First, cut and paste. This DNA will be cut from here. Then it will be inserted into some other DNA, like यहाँ पे insert हो जाएगा. This is cut and paste. Second is copy and paste. So this transposable element will create a copy. And then this copy will be inserting at the right location. So two types, self-transposition, right. So this segment goes and simply gets inserted. Second is called replicative transposition. So first this one will replicate and the replicated copy will insert. So this is cut and paste, ये अपने source से cut होकर और यहाँ पे paste हो जाता है, and replicative one that it will be replicating itself and then it will be entering. So now how many types of molecular recombination are you aware of? You are aware of three types of molecular recombination. Crossing over, which is going to be very much a reciprocal process. But these two processes are non-reciprocal. One, which is site-specific. Second is catalyzed by the enzyme that is known as transposase. But both these processes, but please remember that both these processes happen to be non-reciprocal, that is, it is a one-way transfer. And any molecular recombination, absolutely any molecular recombination, is going to change the syntenic relation. Are we all clear? Is there any question? Formation of GMO can be an example of one-way recombination. It is an example, but that is experimental recombination. It is not a natural recombination. This is experimental recombination. But you are absolutely correct. Any kind of GM organism, it could be GM bacteria, or it could be GM plant, it could be GM animal. Any of this will be a one-way recombination. But in this recombination, experimental processes insert the gene of interest. Vasu is asking, sir, non-reciprocal occurs between non-homologous chromosome. कहीं भी हो सकता है, बैक्टीरियल डीएनए में भी हो सकता है. Explain once transposition. I will ask you to explain what did you understand, and then I will fill the gap. That is a good way to do the doubts. Okay. So Shiva, please unmute yourself and explain to me what did you understand by transposition. Go ahead, Shiva. Yes, Shiva, I can hear you.
So actually, I think the transposition is just like replication of a gene and insert
Okay.
some other genes.
Insert in some other DNA, not necessarily inside of a gene. It can be outside of a gene. Okay. And the first one is that it will not replicate itself. It will itself go and insert. You understand? So that is cut and paste. It will cut itself from a source and it will go and insert into some other DNA.
Okay.
Okay. Maz is asking, transposon are jumping gene? Absolutely. Shraddha is asking, I'm a bit confused. Can you give a quick recap? Recap of what, Shraddha? How is translocation different from molecular recombination? Okay, good question. Translocation is generally a process where there is an accidental breakage of chromosome segment. Means there is no specific enzyme which will come, which will cut, and which will move it to a new location. So there is accidental breakage, number one, and then random fusion. It may fuse, it may get degraded. But if there is breakage and if it gets to fuse with some other chromosome inside of it or at the tip also, this will be known as translocation. So translocation is an abnormal process, whereas site-specific recombination, or for that matter transposition, are pretty much a specific enzyme catalyzed reactions. Okay, Shraddha, किस चीज को recap करना है? Tell me quickly so that I can recap it. दैट्स टू लॉन्ग. उससे अच्छा तो वीडियो देख लेना दोबारा. आफ्टर न्यूक्लियर रिकॉम्बिनेशन मैंने कितना पढ़ा दिया. राइट. सो आस्क मी अ स्पेसिफिक क्वेश्चन. उतना मैं अब रिकैप नहीं कर रहा हूं. ठीक है. मे बी जस्ट द नेम्स. जस्ट द नेम्स क्या? क्रॉसिंग ओवर विच इज रेसिप्रोकल, site specific recombination and transposition. In site specific recombination there is one way flow, right? In transposition it is also one way flow, but there is an enzyme called transposase, and in site specific recombination it goes and inserts into a specific site. Example is lambda phase genetic material inserts in the bacterial genome between gal and biotin operon. So that's what it is. Okay. So sometimes you will indeed need to watch my lecture again. So that will help you, but don't watch the entire lecture all over again. Otherwise, it will waste a lot of time. Is it necessary that it will insert between the other gene? Can it be added? Uh, which will insert in between the other gene? Transposition or site-specific recombination. Site-specific, as the name indicates, is always between the sites. Anshu. Okay. Now we are at homologous recombination, right? So we are now getting closer to crossing over. So here we are. Homologous recombination has another name, and the name is general recombination. Okay, so whenever you hear the word general recombination, it means homologous recombination. Homologous recombination, it always involves the exchange. What would it mean? It is reciprocal process. And whenever it happens between homologous regions. What is homologous regions? Homologous regions are having very similar, actually nearly identical sequences. So when two homologous chromosomes are recombining with each other in crossing over, so mind it, their sequence is extremely close together. The only thing is one nucleotide here, one nucleotide there. Because of that, they are two different alleles. Because of that, they are two different alleles. But at the end of the day, they are having highly identical sequence. Next is in diploid eukaryotes, this commonly occurs during meiosis when the homologous duplicated chromosomes line up in parallel in the metaphase and non-sister chromatids exchange equivalent section by crossing over. Wait a moment, there is something wrong. Not in metaphase 1, but in prophase 1, right? In prophase 1. And also in the prokaryotes, although they don't have meiosis, but there also homologous recombination takes place by REC-BCD pathway. How it will take place? So in a prokaryotic cell, it is possible that from outside a segment of DNA has entered. So this is a segment of DNA that has entered from the outside, okay? And if this segment is highly similar to some part of the bacterial chromosomal DNA, so between these two parts, there will be homologous recombination because the enzymes which are responsible for homologous recombination are sensitive to they show sensitivity to sequence homology. So the moment two homologous sequences come together, these enzymes will get active. So oftentimes bacteria can take up some foreign segment of DNA from outside from their medium, and this will show homology to some sequence that the bacteria are already possessing. But if that is the case in the bacterial cell, it will trigger homologous recombination. And this homologous recombination is going to do what? This homologous recombination is going to exchange the segment. So it is a reciprocal process. Now we are going to focus our attention on crossing over because on crossing over, there are questions. So first and foremost, there is cytological, that is cell biological basis of crossing over. So take a look here, something that we already know. So one chromosome possesses dominant A and dominant B allele, red one, and the other homologous chromosome possesses recessive A and recessive B allele. They both go through replication. Now there are two sister chromatids for every chromosome. Then during prophase 1, during crossing over in prophase 1, segments of non-sister chromatids, so this chromatid and this chromatid, they are non-sister. So segments of non-sister chromatids are exchanged. So notice here, now recessive A has come on this one and dominant A has gone on this one. And when meiosis 1 and 2 are completed, so two of the gametes will be like parent. So this gamete which has dominant A and dominant B is like a parent. This gamete which has recessive A and recessive B is like a parent. But do you notice that this is the recombinant where recessive A is with dominant B, and this is also a recombinant where dominant A is with recessive B. Why did it happen? They are linked genes and they can go through recombination only by crossing over. If the genes are not linked, their recombination does not need crossing over. Their recombination simply needs independent assortment. So this is the cytological basis of crossing over. Sometimes in the examination, you are actually asked the cytological basis of crossing over. So this is your cytological basis of crossing over. Okay. Now we are going to elaborate the steps in meiosis prophase 1, which are closely intertwined with the steps of crossing over. So this is. Now if you see, so meiosis prophase 1 is divided into 5 subparts: leptotene, zygotene, pachytene, diplotene, and diakinesis. So during the leptotene, what happens that, wait a moment. So during the leptotene, chromosomes are dispersed. अभी कंडेंसेशन उतनी नहीं हुई है, but homologs tend to clump together, means अब पास आने की प्रोसेस शुरू कर देते हैं. And this pairing is called synapsis. And this pairing is held together, homologous chromosome की जो pairing होती है, that is held together by synaptonymal complex. So synaptonymal complex, which we call as SC, proteins begin to assemble. Now the next step is called zygotene. Zygo means pair. From here comes the word zygote. Means two things fuse together, therefore they give rise to something known as zygote. Okay. So zygotene. Okay. So here homolog pairing begins and it also gets completed. Homolog pairing begins and it also gets completed. All right. By synaptonymal complex. So pairing is completed by zygotene. इसीलिए इसको जाइगोटीन कहते हैं. But still the chromosomes are not fully condensed. Then comes pachytene. Pachy means a little thicker. So now the condensation increases a little bit. Synaptonymal complex now causes crossing over to take place. So pachytene is the step when there is peak of crossing over. ठीक है. So crossing over की beginning step pairing के साथ शुरू हो जाती है दैट इज ड्यूरिंग द जाइगोटीन, और फिनिश होती है डिप्लोटीन में काजमेटा के डिसॉल्व होने से. सो द नेक्स्ट स्टेप इज सिनेप्टोनिमल कॉम्प्लेक्स फॉर्मेशन. So next is synaptonymal complex causes crossing over to occur. That is reciprocal DNA exchange between non-sister chromatids of homologous chromosomes, or simply homologs, right? And at this stage, chromosomes thicken somewhat, means there is some degree of compaction. So chromosomes thicken somewhat at this level, okay? And then chiasmata. Crossing over is completed. Junction points are still present between homologs. Junction points, ये देखो junction point है, ये वाला junction point है. So junction points still present between homologs. These junction points are known as chiasmata. Chiasma is singular, chiasmata is plural, right? So chiasmata, they denote the site of crossing over. जहाँ क्रॉसिंग ओवर हुई है उसको वो बताती है. सो डिनोट द साइट ऑफ व्हाट, डिनोट द साइट ऑफ क्रॉसिंग ओवर. All right. And finally, dikinesis, homologs move apart from each other. That is dia. Dia means away. Kinesis means moving. Right? So dikinesis. So I have written you the explanation. So when you are writing about the process of crossing over, this diagram with this explanation must be coming. ठीक है ना? So that is an essential thing. After this, there will be a diagram for synaptonymal complex. So basically speaking, synaptonymal complex has plate-shaped structure. Synaptonymal complex has plate-shaped structure. So there are two plates. I'm just showing you a part of synaptonymal complex, not the entire synaptonymal complex. All right. So yes, so now they look pretty comparable. ठीक है? So I'm just showing you a part of synaptonymal complex. So these are two plates, and between these two plates there is something called recombination nodules. So recombination nodule is filled with enzymes which cause crossing over to happen. Right, so these are recombination nodules. And there are many recombination nodule in this one part of recombination. Synaptonymal complex is a very long, like a railway track, a parallel plate system, right? And between these two parallel plates there will be many recombination nodules. For the sake of simplicity, I am showing just one recombination nodule here, and these two recombination nodules will be connected to each other, okay, with these kind of proteins so that they remain in their own place like this. Okay. And then on the two sides of it, chromatin material is attached. So ये एक homolog का chromatin material है, right? And this is the chromatin material of other homolog like this. So now time to label it. So this is chromatin material of one homolog, and this is chromatin material of other homolog. Okay. Plus these are called lateral plates. These are lateral plates. This is recombination nodule. All right. This is recombination nodule, and these are called connecting elements. And within the recombination nodule, there are going to be what? There are going to be the enzymes causing crossing over. And what is the effect of crossing over? Crossing over causes recombination of linked genes. As a result, newer types of offsprings. So novel offsprings even for linked genes. अगर क्रॉसिंग ओवर नहीं हो रही होती, so obviously the newer combination for linked genes wouldn't have arisen. And second thing is that crossing over also has a role in DNA repair if there is damage. Also has a role in DNA repair if it is damaged. So we don't need to delve deep into this, but these are the two principal effects of crossing over, right? And in linkage, if you remember, we have done something called the relation between crossing over and linkage. All right. So why not I copy this page, and in recombination, I just insert this page so that here also you can access this. So that is all about linkage, linkage mapping, and crossing over. So we are done with this. And generally the examination does not ask so much about recombination in general. It is more interested in crossing over. So you very well know the process of crossing over and what is its impact on linkage. So that is pretty much all that we had set out to do. And tomorrow and day after, it will be sex determination, evolution of sex chromosome. And now that we have already studied linkage, so we are going to see sex linkage, sex influence traits, sex limited traits and all that. So tomorrow and day after. And please note the time. It is 9 a.m. in the morning tomorrow and 9:05 in the morning, day after tomorrow also. So उसके बाद we will be done with transmission genetics, and उसके बाद तो mutation वगैरह है. So that is something which is a part of molecular genetics.